Welcome to the Deep Dive, your shortcut to getting up to speed on some fascinating research. Today, our mission is pretty focused. We're digging into a really important scientific paper. It reveals the systemic, kind of historical imbalance that underpins how we even know about global biodiversity. Yeah, we're looking at the hidden biodiversity knowledge split in biological collections. It's led by Gabriel Nakamura, along with a huge international team, actually. And the core idea here is pretty powerful. It's basically saying that the fundamental physical things we need the specimens to define, classify, and ultimately protect species, well, they aren't spread out evenly across the globe. They don't just state the problem, they really quantify it. They call it this knowledge split, and they use hard data.
Okay, yeah, let's unpack that. Because we're talking about something more than just, where museums happen to be located. This imbalance, it really acts as a huge barrier, an ongoing one, for scientific progress. And particularly for researchers working in the places with the most biodiversity. The study makes it, well, painfully clear, really. The actual reference materials you need to study, say, a local ecosystem, they are overwhelmingly housed thousands of miles away from where those species actually live.
Right. And to really get the gravity of this, we need to start with the basics, the kind of fundamental currency of classification. That's the name-bearing type specimens, or name bearers, as the paper calls them. For anyone not familiar, what exactly are these specimens? Why are they so important that a whole paper maps where they are?
Well, name bearers are the absolute fixed reference point for a species identity. You could think of them almost like the legal deed for the species name. They're required by the International Commission on Zoological Nomenclature, the ICZN. And they're pivotal because taxonomy itself, the whole science of classifying life, it's built on them. And this is incredibly crucial when you think that, maybe 80% of the species currently living on Earth haven't even been formally described yet. An estimate, of course, but a huge number.
80%, wow.
Yeah, and what's fascinating here, especially now with all the talk about modern tools like, DNA barcoding or digitization,
Yeah.
direct examination of the original physical specimen, it's often just non-negotiable. For most organisms, you still need that physical proof.
You can't just use a picture or sequence data.
Not always, no.
Yeah.
And think about the urgency. The paper mentions that about 40% of the 562 tetrapod species we consider lost, meaning not seen for 50 years. They're known only from their holotype, that single original name bearer. That one specimen is the only link we have to defining what that species even was.
That really puts it in perspective. And the Nakamura study, they chose a specific group to illustrate this split: freshwater and brackish fish. Why fish?
Well, fish are the most species-rich group of vertebrates, but they're also kind of plagued by huge taxonomic uncertainties. There's just so much complexity. So given the sheer number of species and how tricky classification can be, assessing the name bearers is this constant critical challenge. If you can quantify the knowledge split in this group, it gives you a really powerful proxy for what's happening in biodiversity research more broadly.
Okay, so it sounds like mapping this whole historical flow must have been a massive job. How did the team actually go about quantifying something so spread out and historical? What was the method?
Oh, it was definitely a colossal data effort. They identified, get this, 20,246 fish binomials. That includes the valid species names plus all their synonyms, using Eschmeyer's catalog of fishes, which is the standard reference. And for every single one of those specimen records, they meticulously noted down the source country, so where it was originally collected, and the housing country, meaning where that biological collection is actually located today.
And critically, they didn't just map the geography, did they? They brought in the World Bank classifications: global north versus global south.
Exactly. That classification is really key. It frames the whole issue not just geographically, but socioeconomically. It lets them analyze how historical factors and factors related to infrastructure and wealth have actually driven this imbalance over time. And then they tracked the movement, or rather the location, of these specimens in 50-year intervals, so you can see exactly how the distribution shifted across the centuries.
And to measure the inequality itself, they came up with two really neat metrics that shine a light on the problem. You mentioned one, the endemic deficit. Can you explain that a bit more?
Sure. The endemic deficit, it's basically a measure of a country's, let's call it local knowledge sovereignty. It's a proportion of a country's known endemic species found only there, whose name bearers are actually missing from collections within that same country. So a high deficit means that country has to rely on institutions somewhere else, often far away, just to confirm the identity of species living right in its own backyard.
Okay, that's clear. And what was the second metric? How do they work together to show the split?
The second was non-endemic representation, and this one's simpler. It's just the proportion of a country's entire collection that's made up of species not native to that country. So when you put these two together, you look at a country with a high endemic deficit, maybe in the global south, and compare it to one with high non-endemic representation, often in the global north. You see this really clear, almost one-way flow of biological materials historically.
And the results, I mean, when you see the numbers, they're pretty staggering.
They really are. The big picture finding is that even though the majority of name bearers, over 13,000 of them, were originally collected in countries of the global south, the global north collectively houses 79% of all known freshwater fish name bearers. That's over 16,000 specimens concentrated there.
79%. That's just a massive consolidation of this foundational knowledge.
Right.
Did the study show when this accumulation really peaked?
Yes, and the historical patterns directly mirror, well, the colonial era and its collecting practices.
Yeah.
The dominance of Europe, Central Asia, and North America in housing these specimens. It peaked dramatically between 1900 and 1949. In that period, nearly 90% of name bearers collected anywhere in the world ended up housed in collections within those regions.
90%. Wow.
Now, there had been a slight shift more recently. Since 2000, there's been more of a trend for countries to keep their own name-bearing specimens locally when they're newly collected. But, and this is a big but, global north institutions are still acquiring significant material from elsewhere. Even in the 2000s, 37% of the new specimens housed in the global north were actually collected in the global south.
And the reverse flow, north to south.
Almost negligible. It was something like 0.2% of new specimens housed in the global south came from the north. It's still overwhelmingly a one-way street, historically speaking.
Let's bring it down to those metrics at the country level because that's where it really hits home. The endemic deficit, you said, is practically zero in the global north. Great Britain, 0%, the USA, 5%. But the average deficit for the global south is 88%. Places like Benin are at 100%, Ecuador, 81%.
Exactly. And this is where that structural barrier just becomes so visible. If Benin has a 100% endemic deficit, think about what that means for a scientist in Benin. They need to confirm the identity of a fish species found in their local river, right? It means they likely have to apply for international loans, navigate complex customs procedures, maybe secure visas, or even travel across continents, just to look at the definitive reference specimen for a creature that literally lives just outside their lab.
It's a huge practical hurdle. And conversely, that non-endemic representation metric paints the mirror image, doesn't it? Great Britain and Germany are both at 99%. Their collections are almost entirely made up of specimens collected somewhere else.
Right. And that leads directly into the question of why. What's driving this? The study found two incredibly strong correlations for where these name bearers end up concentrated: GDP, gross domestic product, and native species richness.
Okay, so money and biodiversity.
Essentially, yes. Wealthier countries, the ones with high GDP, tend to have a very low endemic deficit and a very high representation of non-endemic species. The money, the infrastructure to house these things properly, those seem to be critical factors.
But what I found really telling was what didn't correlate. They tested years since independence, right? Thinking that maybe former colonies would have regained control over their collections over time.
And it just wasn't a significant factor
at
all, which is a really critical insight, I think. It strongly suggests that the political sovereignty gained sometimes decades ago hasn't really translated into scientific sovereignty in this context. GDP, the current economic power, that remains the dominant predictor of who holds the knowledge. It underscores how the legacy of those colonial-era collection practices is still deeply embedded, largely unaddressed by structural changes post-independence.
So this split isn't just some interesting historical pattern. It has really profound, ongoing, and damaging consequences for science globally. If you're a researcher in the global south, like you said, you face these huge administrative, financial barriers just to do basic taxonomic work on your local species.
Precisely. We're talking massive travel costs, complicated visa applications, incredibly lengthy international loan procedures.
Yeah.
It can delay the cataloging and description of species by years, easily. And this knowledge gap, it's most severe right where biodiversity is highest in tropical countries, which is exactly where we need the most scientific capacity to understand and conserve it.
And this imbalance, it's not just a local problem for those researchers, is it? The paper connects it to something broader, the latitudinal taxonomy gradient. Could you explain that link?
Yeah, the latitudinal taxonomy gradient, or LTG, it's this observed pattern where tropical species are systematically less likely to be accurately classified, described, or even recognized compared to species in temperate regions. And the paper argues this knowledge split is a likely contributor. because the essential reference materials, the name bearers, are concentrated so far away from the tropics,
Yeah.
where most new species discoveries are happening. It inevitably leads to biases, biased inferences in ecology, in conservation planning. I mean, if you can't confidently identify a species, how can you effectively protect it?
That makes perfect sense. And then there's the whole ethical dimension, isn't there? The fact that material still flows predominantly from south to north even today, it just perpetuates that colonial legacy. The paper mentions concerns about parachute science.
Right, parachute science, where foreign researchers might come in, collect materials sometimes with fairly limited local collaboration, and then just take it home, and that home institution becomes the final repository. And the study even hints that sometimes this can stray into questionable territory, mentioning cases of potentially illegal sampling, particularly in fields like paleontology. Now, some countries, like Brazil, have put regulations in place requiring holotypes of newly discovered species to be kept locally. And that has shown some success in increasing retention. But these are country-specific measures. They don't really address the huge historical deficit that already exists.
So when we think about solutions, the first thing people often jump to is technology. Digital catalogs, 3D scans, technocratic fixes, as the paper calls them, but they're pretty clear. While digitization is valuable, it's not a magic bullet. Why can't a digital copy just replace the physical specimen entirely?
Well, for many types of organisms, certainly including the fish they studied, it's just not sufficient. Digitization is great for label data, maybe surface images, but the really crucial diagnostic features, they often require either destructive analysis, which you can only do once, or highly complex non-destructive methods. For fish, you often need to see internal structures, like tiny bones. That means high-quality 3D imaging, like CT scans.
Which is expensive and needs expertise.
Exactly. It's expensive technology, needs highly trained people to operate and interpret. Not all collections, especially smaller ones or those in less wealthy nations, can afford or sustain that level of infrastructure. And there's another concern, too. The digitization might just end up replicating the existing power structure. If the big global north collections digitize everything first, and they control access to the data, or maybe the quality and resolution of the 3D models, they essentially retain control over the primary data anyway. It could leave the global south still reliant on someone else's digital infrastructure.
Okay, so if technology isn't the complete answer, then the solutions have to be more structural, right? The paper outlines two main paths forward. The first one sounds, well, complicated. Repatriation.
Repatriation, yeah. Returning representative name bearers back to their countries of origin. It's often seen as the ideal solution from an equity standpoint, but it requires enormous political will. You're talking lengthy, complex negotiations between countries and institutions. You need public pressure. And really critically, you have to ensure that the institutions receiving the specimens back have the necessary infrastructure, climate control, security, scientific staff to properly curate and maintain that material long term. Without that capacity building alongside it, repatriation could actually risk the specimens. It has to be done thoughtfully.
So that's the long game, perhaps. What about the second option, which sounds maybe more immediately feasible, facilitating access?
Right. This focuses on bridging the gap without necessarily moving the specimens, at least initially. It involves multilateral organizations, maybe like the Convention on Biological Diversity, actively promoting and providing funding. Funding specifically to cover the travel, the research costs for scientists from the countries of origin to visit the collections where the name bearers are housed abroad. The paper suggests this explicitly giving preference in funding to researchers working in the country where the material was originally found. This lowers the barrier for the individual researcher, even if the specimen itself stays put, for now.
And looking even bigger picture, it seems like our current international agreements around biodiversity kind of miss this whole point.
They absolutely do. You look at things like the Nagoya Protocol on Access and Benefit Sharing. It focuses almost entirely on regulating access to genetic resources, DNA sequences, basically. It largely ignores the fundamental importance of housing the physical reference materials, the name bearers themselves. Without having those specimens accessible locally, scientific development is still fundamentally held back, no matter what rules you have about DNA. It really highlights a need, I think, for narratives within museums themselves to evolve, to move beyond just celebrating, intrepid Western explorers and start openly acknowledging the often troubled social and historical context tied to how many specimens were acquired in the first place.
So wrapping this up, this study, using fish taxonomy as its lens, gives us really definitive quantitative proof of this hidden biodiversity knowledge split. It makes it clear that historical socioeconomic factors, particularly current GDP, are still dictating who holds the keys, the physical keys to understanding life on Earth. And that severely hinders our collective ability to actually understand and protect global biodiversity.
Yeah. And that really leads to the final, maybe provocative thought we want to leave you, the listener, with today. Given that this imbalance is demonstrably real, and it continues to skew our fundamental understanding of life on this planet, what active material responsibility do global North institutions, museums, universities, collections have to actually dismantle these historical legacies? What do they need to do to ensure a truly equitable and ultimately more effective scientific future for everyone?
It's a vital question, isn't it? Especially for those institutions that hold these incredible, irreplaceable collections. Something to really think about. Thank you for joining us for this deep dive.